So hello guys, today we start the topic of respiratory system embryology. Recently you got histology lecture of the the structure of a respiratory system. You had physiology video of this. So now and you had already anatomy class of respiratory system. So now I want to conduct you the essentials of embryology of respiratory system. So please follow the screen. Here we go. So I want to start with general embryology from the beginning after the fertilization. So let's start from beginning. So after the fertilization, I remind you. So after fertilization, we got an embryo. So I mean zygote. So which is a connection of spermatocyte and oocyte. It is present in ample of fallopian tube. It is composed of zona pellucida and zygote itself. But and here after this we start this video. So 1 minute. Sound off. So here we see here fertilization happened in ample and fallopian tube, and here we have zygote. Around it we have zona pellucida, which were preservative for oocyte against our own immunity system. Then follows the process known as cleavage, and in the cleavage you see that from one zygote forms two blastomeres. They are totipotential stem cells from which every cell of human body can be performed. And then cleavage continues, as well it continues, so we see 4-cell stage. So after 2-cell stage we have 4-cell stage. They are also totipotential cells, and they are absolutely equal to each other, so they are different size, and they keep the same size.
Sir,
Yes.
sir, there is nothing there is nothing shown in the screen, sir. There is only blackness.
Again, let me show it again. Thank you for mentioning it. Okay, due to technical issues, so we'll start again. Sound off, mute, yeah. Okay, let's see. Cleavage and blastocyst formation. So again, I repeat the same thing again. So here is the zygote. It was covered with zona pellucida, which were present. It's a hard material, so which were made by ovaries, and then it's in ovaries. It protects the zygote from injurious stimuli from I don't know, might be our own immunity or from hormonal influences. So it has a lot of functions, and we'll discuss it in the female reproductive system. So what the role of the zona pellucida, but still just keep in mind that we have very hardened tissue, so material around this zygote and then around the embryo, which is known as zona pellucida. It also was protective against sperm cells. So before the fertilization, and only it allows only one sperm inside under the zona pellucida, and also we'll discuss it, I guess, in female and male reproductive system histology and embryology. Okay, then follows, and this fertilization work happened in ample of fallopian tube, as it was mentioned before, and then in 30 hours, as shown here. So this zygote is divided by process named as cleavage into 2 cells. This is named 2-cell stage. Following this, of course, a name of the cell is blastomere. Each cell after division is blastomere. It's named as totipotential stem cell, as I mentioned before, because it has potency to get every cell of our body. Every cell, absolutely without exception, every cell of our body is made of these cells. So then 4-cell stage, and you see very slightly in 5-6 days this embryo following the fallopian tube and zona pellucida. So what's the role of zona pellucida here? It actually prevents the expanding growth of this embryo, and you see size of embryo in this stage, in stage of morula, or 8-cell stage, or in the morula stage. So it's same size as a zygote was. So same volume. and it's necessary to not stuck in a fallopian tube. So if it will be larger, it will expand it. So we have extrauterine pregnancy, extrauterine pregnancy, and tubular pregnancy. So might be heard about it. So it's a very hard condition for females. So let's continue. So this is when 16 cell stage happens. It's named as morula, and in this morula stage, in a third day, it enters into the uterus. And in 16 cell stage, all cells are equal. All cells were equal, but then starts the process known as differentiation of cells. So this differentiation happens due to due to, just imagine. So we have 16 cells. Each cell has an approach to the outer. Each cell so is equal in size, and they have approach to central part and peripheral part in the same way. But later, so appeared this differentiation because some cells appeared on upper side, some cells appeared in inner side of this morula, and here starts the process of differentiation, and appears the two cell populations. Two cell populations, they are known as outer cell mass and inner cell mass, and it is or another name of these cells are embryoblast, which is known as inner cell mass, and outer cell mass, which are known as trophoblast. Yeah, outer cell mass known as trophoblast. And recently, so in 4.5 days, so appeared the blastocyst cavity, which is composed of just liquid which is absorbed by this embryo. So and this, let me remind you, zona pellucida, which is present around, it's semi-permeable. It's a little bit permeable for liquids, and can be absorbed, which can be absorbed in a uterine cavity. So you see here absorption of compounds happening, and then this liquid, extra liquid, which appeared here. So they are collected in blastocyst cavity. So as well, it's present in blastocyst cavity. Pressure inside this embryo increases. You see, it's hardened tissue around. Yeah, so hardens material around it. So like by structure, zona pellucida resembles the cartilage. But now pressure is increasing, and actually also some trophoblast cells, they produce enzymes which can very slightly erode this part of zona pellucida, which follows. So now, which follows with disappearing of this zona pellucida, which is known as. So let me remember it. So 1 minute. So it's named as hatching. So in analogy to egg broken. Yeah. So this when eggshell shuts down. So it's same process named here. It's hatching. And now you see, of course, it restricted the quality of. It restricted the opportunity of the cells to stick to something. Yeah. So, and as I said before, so a little reference to pathology. So when hatching occurs in a fallopian tube, so it leads to extrauterine pregnancy. But in uterus, so when it sheds out in uterus, when hatching happens in uterus, so it leads to absolutely normal implantation process. So, and take a look here. So here it approached to the endometrium. I hope you already know what is endometrium. It's a mucous membrane, so we can say it's a mucous membrane of the uterus, yeah, and with epithelia and adjacent connective tissue around. So you see in day 5, so actually in day 5 to 6, so it can be attached to the uterus. So you see we had a migration from this to this. Okay, next part. So okay, it reminds you. So what's happening? So this blastocyst formation, inner cell mass, outer cell mass, zona pellucida still present. Then process of hatching happens. After the hatching, blastocyst attached to the normal area of implantation of endometrium. Yeah. So okay, and when implantation happening, so what happens here? So take a look. We have trophoblast again. So this is blastocyst. Yeah. So from morula forms the blastocyst. When blastocyst cavity is visible, so it's named as blastocyst. So if more than 32 cells are present in embryo, its name is blastocyst. So, and here attachment of this blastocyst happens with the cells which are adjacent to embryoblast. So here we see embryoblast, here we see trophoblast, yeah? And these trophoblastic cells, which are influenced by embryoblast, they are able for another step of differentiation. Then these trophoblast cells, they can perform new cell populations, which are known as, so 1 minute, it's known as syncytial trophoblast, and here it's visible, yeah? So what happening? So actually from these cells appeared the syncytial trophoblast. What means word syncytial? Syncytial means it's these cells, they have no cell division. So here, for example, let's take a look here. So these epithelial cells, they have borders. Here one cell is distinguished from another cell. So same we see here. So one cell is distinguished by another cell with this cell membrane. But in syncytial trophoblast we have a lot of nucleuses, a lot of individual cells, but with fused cytoplasms. So this is known as syncytial. This portion of word is syncytial, means this. So and everywhere, everywhere in future you listen syncytial something. So it means the syncytial cells means they have fused cytoplasms. A lot of nucleuses in one cell, so in one cytoplasm present. So this is syncytial trophoblast. So and this population of cells, syncytial trophoblasts, they are formed from this portion of trophoblast, and now to distinguish them one from another, we name this as cytotrophoblast. The syncytial trophoblast and cytotrophoblast. So cytotrophoblast now remains same as trophoblast. Syncytial trophoblast with all these properties we had before. And this syncytial trophoblast, they have absolutely very specific property. It has expanding growth. It has a lot of enzymes, proteolytic enzymes, which can erode the uterus. They can invade very deep in a connective tissue, which is present here. Due to this, it drone this embryo and implant this embryo to the endometrium. So, and in endometrium, so which process happen in endometrium, we'll discuss it in female reproductive system, but you have almost understand, yeah, that uterus is preparing every menstrual cycle. Uterus is preparing for implantation. So it prepares for implantation, and that's why here we have a lot of glands which produce a lot of nutrients for this embryo. In other hand, it produces a lot of blood capillaries here, so the spiral arteries might be you remembered from previous topics. And then embryo is implanted here. So then what happens inside? So syncytial trophoblast keeps doing its function, yeah, so it invades and spread all over the endometrium here, and and a little spoiler for you, in future it will help to form the organ which you heard about. It's placenta, yeah, it helps to form placenta. So but yeah, I'll explain it a little later. But inside, what we have inside it? So embryoblast, we forgot about embryoblast, yeah. So here blastist cavity is still present, but here we have embryoblast. Embryoblast here goes into another form of differentiation. Another form of differentiation when we have two cell populations from embryoblast. It forms epiblast and hypoblast. So epiblast and hypoblast. So and usually it's not blue and yellow, of course, but just for memorizing. So it's always in all books it's shown as blue and yellow cells. Because in further you can confuse them. So what's the future of them? So we'll discuss very soon. So, but it is future embryo. So actual future embryo is made of these cells: trophoblast, cytotrophoblast, and syncytial trophoblast. They are more responsible for invasion only, and it will be and all cells which are present outside. So which is the main cell populations here. Yeah. They gonna be eradicated, and they gonna be atrophied, apoptosed, or they will form the placenta. So in future. So here we see. Then this embryoblast forms these structures. So you see epiblastic cells, these blue cells, epiblast cells. Please remember it. They form the roof of their cells, of this structure, roof of epiblast. and cavity which appears inside this epiblast cells, between this epiblast cells and amnion cells, it is known as amniotic cavity. Amniotic cavity. So might be you heard about amniotic cavity from from any pregnancy. Yeah, as I said, amniotic fluid. You heard about it. Yeah, and actually this amniotic fluid which covers entire embryo, so entire fetus. Yeah, in later periods of life, later periods of pregnancy. So actually now it's very small sac here. So but in future it will expand everywhere. So and also today we will show it for you. So say to this guy, so whose name, so 1 minute, and this amniotic cavity is here. Yeah. So this amniotic cavity, as I said, a future develop entire fetus or even embryo. So and what is a hypoblast? We'll discuss it right now. Yeah, you see, from hypoblast appeared this new cell population, which lines the entire trophoblast cavity. Yes, so blastocyst cavity. So near the trophoblast formed these very teeny tiny cells, thin cells, simplest ones epithelial cells, which is known as exocelomic membrane. And the cavity which is found here, it's named or exocelomic cavity, but much better it is known as a yolk sac, the primary yolk sac or primitive yolk sac. So we have a lot of names, so here don't lose the line, don't lose the line. So but anyway, so this is primitive yolk sac. Might be heard about it. It's similar to birds. Yeah, so in a bird egg, so you've seen this yolk sac. What is yolk sac? The yellow portion of egg. So it's a yolk sac. In humans, we also present here, so it also a confirmation of theory, evolution theory. But I keep heard from students that they don't agree with the theory, Darwin's theory. So it's lie. Yeah, so we are creatures. So if you're a creature, but it's not in a way you expected. So it's evolution. It takes place here, and you can see it on this example. We have yolk sacs. We don't have eggs. We don't have to feed it. We can feed our embryos from uterine glands and blood, but still we have yolk sac because evolution has no other options to make embryo. So this is like one of confirmations of evolution theory. Okay, we'll discuss it in the practical class. So and furtherly we can see that that we have two cavities. So please remember these two cavities: this amniotic cavity and yolk sac. And between them we have this bilaminar disc, which is composed of epiblast and hypoblast. I hope you remember. Yeah, so this and this layer membrane made up of two cells is present here. So between once again amniotic cavity and yolk sac, and it will remain for a long time later. So and here mention the syncytial trophoblast. Syncytial trophoblast keeps growing. It invades blood vessels and glands and forms estimated the structure known as lacuna. This lacuna, they are like primitive blood vessels inside the embryo, and now circulation of blood going on in embryo, and then feeding of this embryo fully established now. So it's happening in trophoblast. Yes, the syncytial trophoblast work. So it's outside of the embryo happening this, but what happens inside? From the same syncytial trophoblast, cytotrophoblast here, so this cytotrophoblast, from it produced another cell population which is known as extraembryonic mesoderm. Extraembryonic mesoderms, they are like jelly-like cells. They form the mesh structure of cells here, which present like jam-like structure around this embryo, and they fill all the space between yolk sac and cytotrophoblast. This is known as the extraembryonic mesoderm. So don't confuse it with intraembryonic mesoderm, which in future forms some organs. You heard about it. So this extraembryonic mesoderm is responsible also for formation of placenta, first of all. placenta will be formed from extraembryonic mesoderm, but not the intrahumanic structures. So take a look. Actually, so a little spoiler. So all cells of our body, or for adult bodies, they are derived from epiblast, not hypoblast. So all cells of hypoblast will be replaced by epiblastic cells very soon in a lecture which is following. So and now you see from outer side we have formation of circulation in embryo. From inside it forms extraembryonic mesoderm, and this extraembryonic mesoderm, as well as mesoderm, as well as mesenchymal cells without attachment one to another. I hope you remember we also discussed it. These cells, they have no attachment of their cell membranes. They don't have coherence. Yeah, and that's why very soon, due to production of liquid between these cells, cell masses, we have appears a cavity. And this cavity is known as chorionic cavity. This is named as chorionic cavity because so all structures which are above this chorionic cavity, it is known as chorion, and chorion is a fetal part of placenta. So now we can see that what is the structure of fetal part of placenta, structure of chorion. I remind you, this is green one, syncytial trophoblast, purple, violet one, so it is cytotrophoblast. And this orange one, yellow orange one, so it is named as extraembryonic mesoderm. So together the three, it is named as chorion, and this chorion is a fetal part or embryonic part or fetal part of placenta. And then so another part of placenta is maternal part of placenta. They are made up of endometrium. Okay, let's continue. Cytotrophoblast, syncytial trophoblast, extraembryonic mesoderm, and chorionic cavity. So let's continue. And what happens next? It reminds you what is happening here. Yeah, so forms amniotic cavity, yolk sac, and between them we have bilaminar embryonic disc. So please remember this name, bilaminar embryonic disc. So this bilaminar embryonic disc, so it separates amnion from yolk sac. So and everything will change in gastrulation process. So you see, and gradually yolk sac reduces in size. So it relatively reduces. So it's not reducing actually. Entire embryo is growing. So, but this yolk sac remains almost remains unchanged, but not growing. So and this cavity, which is known as chorionic cavity, increases in size. This cavity increases in size. Yolk sac a little bit reduced, and here forms the secondary yolk sac. Secondary yolk sac. So this is secondary yolk sac. You see, it's much smaller than the primary yolk sac, and here I want to pay your attention for this structure, exocelomic cyst. It doesn't matter at all for us. It has no sense for us. It has no function for humans. But these exocelomic cysts were very important in egg-producing animals. So for reptiles, yes, first of all. So in reptiles, they had exocelomic cyst for its air containing part of egg. So if you ever open the egg, so you've seen this small portion which is the air containing portion there. Yeah, this air containing portion, it is known as exocelomic cyst. It's necessary to prevent drowning of these eggs in water, or also for ventilation of oxygen in this egg. So, but here we have absolutely no function of it, but still is present because we are from same ancestry as reptiles. Okay, and also extraembryonic mesoderm here. So it is connected with embryo with a small stalk, which is known as connecting stalk. And another name of this connecting stalk in future, it will be Maybe someone will is figured out what is the name of this connecting stalk. Who can say it? No one? Okay, it's a future umbilical cord. It's a future umbilical cord. Okay, let's continue. Here follows the next lecture. So yeah, and you see day 12, 13. So these structures are formed. So now let's talk more about embryoblast. So process of syncytial trophoblast invading. So it's following. So in the same way as it was before. So same it continue expansion and inside the endometrium and don't go deeper. So to the muscular layer, so to the myometrium. But if it passes this, so if expansion more radical, if expansion goes to the muscles, so it leads to so anomaly of pregnancy. So okay, we'll discuss it. So I'm confused in terms, but we'll discuss it in gynecology pathology. So. And here you see we have amniotic cavity, yolk sac, bilaminar embryonic disc composed of epiblast and hypoblast. This yellow hypoblast, bluish epiblast. Yeah, amniotic cavity is formed by epiblastic cells and amnion. Yeah, and yolk sac is composed of hypoblast and exocelomic membrane. Exocelomic membrane. And this exocelomic membrane forms the yolk sac inside or exocelomic cavity. Okay, hypoblast, epiblast. Okay, here we understand it. So here we have connecting stalk. So which is necessary furtherly for blood vessel formation here. Okay, never mind. So and now let's imagine it like two bubbles present here. So that's oversimplification of process, but still keep in mind. So now you have to be sure in this process. Here we see amnion. So amnion is a external part. So amnion, you remember, so it's made up of epiblast. So but thin layer, so which will will form amnion, amniotic cavity. And then from downside we have umbilical vesicle or yolk sac and hypoblast. So I hope you clear with this concept. And now we have to define these parts: cranial end and caudal end. Cranial end is a head end. Yeah, so cranium is a head, skull. This is cranial end of embryo. This is caudal end. So now it's not defined yet, well, but soon it will be shown how it's defined. Caudal end is named as a tail end. So this cauda means in Latin. And you see from caudal end. So okay, let's explain these three structures. We have oropharyngeal membrane, future oral cavity, and pharynx. So pretty obvious, yeah. So this is just membrane. It's just like indication of the epiblast. So with the oropharyngeal membrane, and its name is oropharyngeal membrane. Furtherly, we'll discuss what is it. Here we have cloacal membrane. Cloacal membrane. So what is a cloaca? I hope you remember it from biology. If not, so I remind you. So in reptiles, in birds, we don't have separation of genitourinary tract and the digestive tract. They are like thrown in one common system, which is known as cloaca. This cloaca is like for all wastes which are present in the body, so it removed from the cloaca. And here we have cloacal membrane from which it forms. So and one structure which used to be which you used to forget about, and especially teachers of biology in school, they forgot to say about it. It is known as primitive streak. So primitive streak is a little groove with this round end in the end. So which very important to next process, following process known as gastrulation. Here let's see it. in a cross section of this embryo, so it will be more visible. So this primitive streak, so this is groove of primitive streak, starts the process of migration of the cells, migration of epiblastic cells, and they replace the hypoblast. Here you see hypoblast is replaced with the epithelial cell, ectodermal cells. Epiblastic cells replace the hypoblast and now forms the definitive endoderm. So definitely this is endoderm. Not hypoblast is endoderm, but epiblastic cells which were migrated and replaced these hypoblastic cells, they are named as endoderm. This is the first step of gastrulation. Here starts the process of gastrulation. So this is, and here it's over. And then continue the process. So it leads to a process known as epithelium-mesenchymal transition. We already discussed it in the cancer formation. Yeah, these epithelial cells. So here you see they are typical epithelial cells. One cell is attached to another with the help of connexins, different categories, and they have basement membrane or something. And it doesn't allow to it forms a very strict arrangement of the cells. Yeah, but here starts the process of losing of these connections, and in case of pathology in cancer, we discussed that it leads to metastasizing, yeah, of cells. When these connections are lost, and then cells are freely motile. But here it leads to formation of third layer or third germ layer formation. It's known as mesoderm. Mesoderm forms. And these cells of mesoderm now, they are named as mesenchymal cells. Mesenchymal cells, they are named as mesenchymal because they don't have connection. And now we see we have three germ layers. Ectoderm, epithelial layer composed of ectoderm. Then endoderm, also epithelial cells. And between them we have like inner part of the pie, we have mesoderm. So mesoderm is like jelly. So it has no connection to one cell to another, but it has very great potentiality for being differentiated. Yeah. So as well it will be differentiated. So it forms different organs and systems in our body. So on this process, gastrulation is over. So now don't believe in biology books. So they explained for frogs, but we are talking about humans, and this is process of gastrulation. Again, what happened in gastrulation? From epiblast and hypoblast, from bilaminar embryonic disc forms a trilaminar embryonic disc. So bilaminar were composed of epiblast and hypoblast. So, but in the end of this process, for first of all replacement of hypoblast with the endoderm. And second process is the migration of mesenchymal cells between them. So it is an entire this process known as gastrulation. Okay, let's continue. So now very important process. I hope you'll repeat thinking about it. So now we discuss the process which happened in primitive streak. Yeah, in this primitive streak, but in a primitive groove of primitive streak. But here in the end of primitive streak we have primitive node. From this primitive node starts the formation of notochord. So notochord gives rise from it. So take a look here. So you see, primitive streak leads to formation of mesoderm here, yeah, and mesoderm forms around the primitive streak, but rest part of embryo, so present without mesoderm now. And sharing of those mesoderm happens in this process. So you see mesoderm spreading by these roots in between this ectoderm and endoderm now. And this mesoderm present everywhere besides 2 points. This one, it is known as oropharyngeal membrane, future oral cavity and pharynx, and here cloacal membrane. So it's future anal canal and urogenital sinus. So and here between the oropharyngeal membrane and primitive node of primitive streak forms a structure known as notochordal process. Notochordal process is the first axial organ of our body. So it's this is axis of our body. It will be cord on which all our all our axial organs are formed, starting from nervous system. We'll discuss it in nervous system pathology topic. Also all our blood vessels, all our bones. So like our backbone, for example, it's made under direct control of notochordal process. Okay, we'll discuss it soon. And now you see, this notochord is present here between oropharyngeal membrane and primitive node, and ectodermal cells, which are present above the notochord, they form the structure known as neural plate. And that's why might be heard about and was a bit confused that ectoderm, which forms the skin, yeah, so epidermis of skin, actually is responsible for formation of the nervous system. Might be you were confused. I was when I first time observed it. I was confused, but now you can understand why. So we have ectoderm and notochord, which is present here. So it like determines by these their factors which are produced by notochord. They determine the differentiation of ectodermal cells into neural plate, and it features this neural plate folds to form the nervous system. So and formation of nervous system we'll discuss more in a further topic of nervous system pathology. So but still keep it in mind. So now growth of formation of notochord. So let's explain it in a very short way. So you see here mesoderm is forming, so this ectoderm and endoderm now, and primitive pit, so it's a deepest part of primitive node. So the notochord grows in this way. So in the opposite side to primitive streak. So here we had primitive streak, I remind you, and in opposite side of primitive streak towards the oropharyngeal membrane starts the formation of notochord. So here you see notochord is an empty hollow organ, and here it has notochordal canal, and it almost reaches the oropharyngeal membrane, and it has direct connection to amniotic cavity. So here bluish is the amniotic cavity, here yolk sac. And now happens a process which used and we don't fully understand the reason of this process, but okay, let's discuss it. So this tube is fusing with endoderm, notochordal plate intercalated in the embryonic endoderm, so it just fuses with it, yeah. And what happening here? So actually fluid from amniotic cavity now temporarily attached to yolk sac, and researchers they don't fully understand the reason of this process, but one thing happens very clearly. So it's for equalizing of pressure in amniotic cavity and yolk sac. So let's continue. This fusion of notochord with endoderm present only 24 hours, and after this 24 hours it returns back to the notochord tube. But for this 24 hours we have this canal known as neurenteric canal. So not neural canal, neurenteric canal. Neurenteric, so it's from word neuro and enterus, so like brain, and so nervous system and digestive tract. So because endoderm firstly forms the digestive tract. So here you see neurenteric canal. It's a connection between brain and digestive tract. It remains once again only for 24 hours. Following after this refolding, folding back of this notochord happens, and notochord is forming back tubular structure. And now it's detached from both, from ectoderm and endoderm. And then notochord starts to provide this activity to form neural plate around. So neural plate is following here. So and then okay, in urinary tract development we have to discuss it, but now it's not so important. So along with the process of notochord formation, elongation of this endoderm into the umbilical cord, it forms the allantois. Allantois will form the urogenital sinus and anal canal. So information of these structures is very important is followed due to allantois. Okay, let's talk about folding fibrilles so very shortly, and then we'll start the respiratory tract embryology. So now here we see after the gastrulation we have the structures. I hope you remember. It is amniotic cavity, yolk sac, ectoderm, blue ectoderm, then red endoderm, mesoderm, sorry, red mesoderm, and here endoderm. In this side, from this side we have oropharyngeal membrane, future oral cavity, so where ectoderm and endoderms are connected, and from this side we have cloacal membrane. So also ectoderm and endoderm are connected without mesoderm, and mesoderm shouldn't be allowed there. This is allantois, and this is in cranial end of embryo, so which we didn't discuss yet. So forms the primordium of heart. It is known as precardial plate. So this bunch of mesenchymal cells, which furtherly will form all cells of heart. So and you see, it's most cranial part of our body is made up of not brain, not head, yeah. So but here we have the heart. So it's not mistake. So please remember it. Then I'll show you how it appears in the chest. Now, and this process, further process, is named as a folding. So this you see a lot of process going along here, so that why you can be a bit confused for a point, but try to understand. So as I said, notochord so determines the formation of neural plate, and neural plate forms the neural fold, and the neural fold forms the neural tube. Yeah, neural tube. Along with this process, notochord also determines differentiation of mesoderm, which is present on both sides. It forms of so now it will be shown. Okay. Anyway, so three layers of mesoderm are formed, and then following the process of folding. And now you see, amniotic cavity was a very small cavity, but now due to folding process, this amniotic cavity starts enveloping entire embryo, and yolk sac in relation to this, respectively to the growth of growth of amniotic cavity, it starts reducing in size. Now you see, amniotic cavity just almost fully surrounds the embryo, and only a small portion of umbilical cord, a small portion of yolk sac is remained, and it is fused with umbilical cord structures. So now mesoderm, which is presently it's known as lateral plate mesoderm, so most lateral portion of mesoderm, it forms two structures. It is known somatoplevra, which is present near the ectoderm. This mesoderm which is present near ectoderm known as somatoplevra, and this portion of mesoderm which is present near the yolk sac is known as splanchnoplevra. And in between these two structures we have this cavity, intraembryonic cellome. So it forms all three cavities that we have in our body. So these three cavities, like pleural cavity, pericardial cavity, and peritoneal cavities, they are formed from this intraembryonic cellome. So in this, and of course you see that mesoderm of somatoplevra, it is a primordium of parietal pleura or parietal pericardium, and also parietal part of peritoneal cavity. And splanchnoplevra is the visceral part of the same cavities in future. So, but yeah, I'm going too far. And now you see it goes closer and closer. So this is yolk sac. So you see it's connected. It became this umbilical vesicle. So became a part of yolk. It became the part of connecting stalk known as umbilical cord. Okay. So it was in a middle portion, yes. But let's take a little closer to the cranial end. So what happens here? So also the similar folding here goes, absolutely similar process we discussed before. So also mesoderm differentiation, neural groove differentiation, formation of neural tube. So this lateral mesoderm, formation of cavities, yeah. Also expanding of amniotic cavity, and then this amniotic cavity here absolutely cuts down the yolk sac. So on this, in projection of this more cranial portion, So yolk sac is absolutely absent here, and here forms this yellowish structure, which is known as embryonic gut tube. So this is closer to the cranial part is known as foregut tube. So closer to the end, it will be named as hindgut tube, and in the middle, so we discuss midgut. So 3 portions of gut tube. So it's a future digestive tract, and now it will be very important because respiratory portion is made up of this embryonic gut tube, especially from foregut. Okay. And you see, when enveloping of amniotic cavity happens, this splanchnopleura, somatopleura is remained, and splanchnopleura is remained, and between them forms a cavity, entire cavity, and it surrounds the digestive portion, surrounds the embryonic gut tube, and its name is intraembryonic celome now. So development of these cavities we'll discuss furtherly in some other topic, I guess, in the cardiovascular system. I think I'll discuss, or in digestive tract system. So, and now let's see it from sagittal plane. I said it before that here we have, might be someone remembered. So 1 minute. So this structure is known as chorion. Yeah, so chorion was made up of extraembryonic mesoderm, then cytotrophoblast and syncytial trophoblast. Together they form the chorionic villus. So this chorionic villus, so it invades the maternal part of the uterus and gets a feeding from the uterus, and blood vessels started from the chorionic villus. So they constitute connecting stalk and then established the circulation in embryo. So furtherly we'll discuss it a little later. So here we see on lateral view, on sagittal view, we've seen in a cross section. Yeah. So but now let's do it in longitudinal section. Here we have heart, septum transversum. So it's a future. Future diaphragm, and now see folding happens, and heart from cranial portion appears closer to the central portion of our body, and septum transversum, also mesodermal structure made up of mesenchymal cells, appears closer to the middle of the body, so due to a folding process. And the septum transversum, once again, it's a diaphragm, and predominantly diaphragm is made up of the septum transversum. And now you see 3 portions of digestive tract is formed. Is a foregut, midgut, so which is connected with the yolk sac, and hindgut, so hindgut. So these 3 portions of digestive tract. But foregut also, and now we have to concentrate our attention on foregut. So because foregut is the main structure responsible for respiratory tract formation. So, if I'm mistaken, that's it. Yeah, here our lectures. Here we have pharyngeal arches. So, what are pharyngeal arches? So, very fast we'll discuss it. So, here we see that foregut. Foregut forms grooves. So, forms grooves. It's first groove, second groove, arch forms first arch, second arch, third arch, fourth arch, fifth arch, and sixth arch. But fifth one is usually reduced, and that's why we can see only first, second, third, fourth, and then sixth, because fifth one is fused with the fourth. And pharyngeal arch 1 splits into 2. It's mandibular prominence and pharyngeal and maxillary prominence. Okay, we'll discuss it in face formation, so not now. But we have to concentrate our attention on the sixth arch. So this is hindgut. Yeah. Foregut. Here you see this is foregut. This is arch. First, second, third, fourth, and sixth arch. And this is okay. It's a respiratory outpouching. Okay, this is pharyngeal arch, and okay, what's the structure of this pharyngeal arch? They are composed of, first of all, endoderm, ectoderm, mesoderm between them, and and here in each of this pharyngeal arch forms the blood vessel. It's a blood vessel, and then nerve, these neural crest cells. Okay, never mind, and also cartilage. So primordial cartilage is formed here in each pharyngeal arch. So here you see a very strange situation when arch 1, arch 2, arch 3, arch 4 and arch 6, because arch 5 is just fused and not developed in humans. And here you see laryngeotracheal groove. So you see it's a foregut, and it forms a pharyngeal endoderm. And this endoderm lines from inner part. And you see pharyngeal groove is the ectodermal portion. It's known pharyngeal groove. It will be responsible for face formation and neck formation. And pharyngeal pouches, they are responsible for intra pharyngeal structures present in respiratory tract and digestive tract here. The pharyngeal pouches, they have 1, 2, 3 and 4 only. So fifth one is absent. This is known as pharynx. Yeah. So, as I said before, so this mesodermal part is composed of cartilage, nerve, and muscles, and of course aortic arch. It is a mesodermal structure which forms the blood vessel. And pharyngeal groove, pharyngeal pouch. Between them, we have very thin layer of mesoderm. So this oropharyngeal membrane, which is going to be ruptured and then form the oral cavity. In 28th day of embryo development, we see all these arches. So pterygoid diverticulum. So okay, we'll discuss in the endocrine system. And now you see this changing of this pharyngeal arches. So this first pharyngeal pouch, it's a future ear canal. So this is ectodermal part and endodermal part of ear canal. So they form the ear. Actually, this is pharyngeal pouch 1, pharyngeal pouch and pharyngeal groove. Okay, and these portions, they're responsible for thyroid gland and thymus formation. Also, we'll discuss it a little later. Parathyroid glands also formed by this. Okay, and you see, and they're endodermal in origin, but they are incorporated into the structure of thyroid gland, and in a deeper part forms the thymus, but they're endodermal in origin. So that's my thing you have to remember. So they form endocrine gland, but still they had connection to endoderm. And also tonsils are formed here, palatine tonsils. So that's not our portion. So this is thyroid gland formation. But here I want to pay attention to this part. Respiratory diverticulum. This is beginning of our next lecture. So following lecture. This respiratory diverticulum continues and form bronchi. Okay, we will discuss this. And this thyroid gland descending. So okay, here is the endocrine embryology. So you can observe it by yourself if you want. So this also will be discussed. Another topic. So now, yeah, which changes happen in pharynx? Yeah, in pharynx, this ectodermal portion forms the body of tongue without mesoderm, without muscles. Yeah, so all not muscular part of the tongue is made up of ectoderm. Then root of tongue is made up of endoderm. and this terminal sulcus where we have papillas, yeah, this papilla valuated, surrounded by walls. So and here from mesoderm of cartilage of the sixth arch forms the epiglottis cartilages of the larynx. So this is about respiratory portion. So now let's talk about respiratory.
Sir, it's enough today.
It's not enough, so don't complain. We have
Sir,
more
we are
than
going
20
for
minutes
Jimmy,
more.
sir.
So that's why we have today's topic actually respiratory system. Yeah, so that why don't complain, just listen. If you're tired, so you can observe it in the YouTube later. So in four-week embryo, so as we discussed, pharyngeal arches were there. So this endoderm, or known as foregut. This is ectoderm, named as so pharyngeal arch. So, and as I said, below the sixth pharyngeal arch forms a respiratory diverticulum. Respiratory diverticulum, and this respiratory diverticulum, it's as you understand, it's a future trachea. This is pharynx. So let's take a look. And we have 5 stages in development of respiratory system. So and in lecture, in PPT of the lecture I sent to you in Google Classroom, so you can observe so theoretical part of this. So and I'll ask you in practical class about this theoretical part, but just in general I explain here. Hypopharyngeal eminence, pharyngeal pouch, laryngotracheal groove. So this will form trachea. You see. This laryngeal opening, how it's formed. So I remind you, so mainly endoderm is corresponding to digestive tract. So that's why to form respiratory tract, we have to form something extra of this. And now you will see it. Endoderm outpouches here. So, and reaches the laryngeal inlet. So the hypopharyngeal eminence reaches the this inlet. So and here forms the structure which forms the groove, two grooves which form epiglottis, and then it's correlated with the tongue. Yeah, so here we have tongue and root of tongue, which is connected to epiglottis, so which allows us to breathe and stop breathing when we swallow something. Yeah, so this tongue, root of tongue, so called a collapsion of a respiratory portion. Yeah, and that's why we don't choke so many times. So how trachea is formed? So everything starts with this embryonic phase of trachea, embryonic phase of lung development. This is, of course, respiratory diverticulum. It grows along with the intraembryonic mesoderm present. Yeah, this is intraembryonic mesoderm, mesenchymal cells, and endoderm here. Yeah. They play a very important role. These mesenchymal cells which present here, they play a very important role in formation of, in one hand, formation of blood vessels, in second hand, formation of parenchyma of this respiratory tract. Tracheal bud, and you see in tracheal bud it causes the splitting of this. It's a bifurcation of trachea happens, and after bifurcation of trachea forms the bronchial buds, pretty obvious. And then bronchial buds furtherly keep dividing. Yeah, so in right one splits into 3 portions, left one splits into 2 portions, and splanchnic mesoderm so divides into 2, forms the visceral and parietal pleura. You see, right bronchial bud forms 3 bronchi and secondary buds, and forms the left one forms 2 bronchial buds. So it's correlated with the anatomy. I hope you remember. In anatomy, you remember that from in right portion, in right lung we have 3 main bronchi. In left we have 2 main bronchi. And then branching continues. It was embryonic phase, but then when branching continues, so many branches are happened, it will be named as a second stage, is pseudoglandular stage of respiratory tract development. When 16 more branches are happened, they are named as pseudoglandular stage. So let's see. So branching continues. The mesenchyma here allows to form the parenchyma of this organ, and lobes are already like separated from each other, regarding the growth of bronchi. So, and now passaging of air is already possible, but you remember that it's happening in uterine, where it is surrounded by surrounded by water, so that's why air conduction is impossible just because the water is not here. But actually, conductive pathways has to be formed, but which are not necessary to embryo itself. So then, respiratory diverticulum branches here and forms structures which in histology will resemble the gland, right? And that's why this period is named as pseudoglandular period. And here around we have mesenchymal cells, or splanchnic mesenchyme, and capillaries and veins here. Capillaries and veins from the aorta of sixth arch of sixth pharyngeal arch. Then, when branching continues after 16 level of bifurcations, it forms a canalicular period. In this canalicular period, all cells becoming just cuboidal, and blood vessels embedded very deep in this canalicular period. But here we don't have alveoli yet. And this is it forms a terminal saccular period, so it's also just named as saccular period. So when first forms the saccules, which also known as acini. Acini are formed, and in this saccular period form the two types of cells: type 1 pneumocytes and type 2 pneumocytes. So actually, first of all, form type 2 pneumocytes because they're cuboidal cells of alveoli. They're named type 2 pneumocytes, and also Clara cells. Now, but here we have type 1 pneumocytes, which forms a simple squamous epithelium of alveoli furtherly. And this finally we have a fifth phase, so main period. So for us it's alveolar period. And finally forms the alveoli. So this thin-walled alveoli, which allows us for exchange of gases, exchange in part. And it's you see period of time. Which period of time is mentioned here? 32 weeks of fetus and 8 years. So till the 8th year, so this alveolar period is not fully formed. So main difference between alveolar period and saccular period is the formation of surfactant. And we discussed what is surfactant, surface-active agent, which is made up of Clara cells and pneumocytes type 2, and they increase the superficial tension here, and they don't allow to collapsion of the alveoli here. So, when alveolar period is present in more than 30% of lung parenchyma, so a newborn can survive. He can breathe, so it can breathe. But if it's not developed, so it might be in when birth happened before 32nd week. So we have a membrane disease. Membrane disease. It's a fibrin membrane disorder. So you will discuss it in pathology. You will discuss it in childhood pathology, or already discussed it before. Okay. Type 1 pneumocytes, type 2 pneumocytes, and if I'm not mistaken, that's it. I wanted to explain for you. That's it. PPT of the lecture you can find in the Google Classroom. If you didn't get it, so I can share it to your groups. If you don't, so please type me in a group. I will share this PPT there. Okay, thank you for attention. So I'm gonna start
Excuse me, sir.
the presentation.
Class
Yes, you have questions?
Sir, Shafqat Ali not allowed to.
So what Shafqat Ali?
Sir, I am not allowed. I try to join the class, but I am not allowed.
Yes, because you were late. That's why you're not allowed.
No,
Might be.
no, sir. When class start, I will join it.
So might be you did something wrong. So if you followed rules, so nothing would happen. Okay, any questions related to the lecture? So now I'm checking attendance. So attendance check, then in the chat. Don't forget to write your name in the chat, or I won't mention it. Okay, no, if you have no more questions, you can leave. Thank you for attention.
Thank you, sir.